Hello everybody, welcome to Skin Aging Unlocked for the Skin Diary. I'm Fiona Clarke from the Menopause Research and Education Fund, and with me today my wonderful co-host Clare Kiely, who you will know and recognise, who is a consultant dermatologist and has a master's in skin ageing, and is also hugely interested in skin cancer. And our guest today is Beibei Du-Harpur, who's a dermatologist and who's basically devoted her career to research, is a National Institute of Health and Care Research academic lecturer with a PhD in molecular medicine as well. So you don't get much more qualified than that. Beibei, how did you get to this point?
I started my dermatology training in 2016, and during that time an amazing researcher called Fiona Watt was based at King's College London, and she is a pioneering skin stem cell scientist and a pioneer of regenerative dermatology, and she was doing some really interesting research using all these novel techniques to understand skin biology, and I really wanted to be part of that. So I ended up doing a PhD at the Francis Crick Institute, co-supervised by a computational biologist, Fiona, and also Magnus, who's another dermatologist. And then after that, I applied to do this clinical academic post, which I'm currently in, so where I spend half my time doing research and half my time in the clinic seeing patients.
Busy. When it comes to research, I don't think people actually understand what it takes to bring a good evidence-based research paper to publication.
Yeah.
What are the processes that you go through?
So there's obviously lots of different kinds of research out there, and where I'm based, we have more clinical research as well as more basic science research. It spans kind of the whole spectrum. When it comes to establishing an evidence base for clinical research, we're thinking about things such as clinical trials, randomized clinical trials, looking at treatments. That's what we would consider very translational research. And then you have more basic science research as well. And that's about collecting data, generating experimental models, interrogating those models to generate more data and put together a picture so that you can better understand something. So I'm in the early stages of a research career, really. I mean, these careers are often decades long. Mm-hmm. And I'm in the process of building a big research project to better understand some common skin diseases that we see in the clinic that I'm very passionate about. Acne and hidradenitis suppurativa are my sort of main areas of interest. And we are trying to build this big study to understand these diseases much better, looking at lots of different things, both clinical data and demographic data, as well as molecular markers in the tissue, blood, serum, all of that kind of stuff. And we know from the research that has been done in the past in conditions such as psoriasis that these huge cohorts of patients who have donated their data has empowered us with so much knowledge over time. And the process by which you can gather this data is very long. You know, you have to apply for grants, which are peer reviewed, and you're grilled and assessed very thoroughly to be funded to do projects. Feasibility checks are very much a part of that. You have to go through an ethics approval process in order to recruit patients. You have to make sure that the study that you're doing is acceptable to patients and so on. So it's a very long process, and people often ask me, what do you do in your research time? They imagine that I'm busy in a lab bench pipetting or something, but actually most of the time I'm writing. So you're writing those ethics proposals, you're writing those grant applications, you're writing papers, you're peer reviewing, which is again more writing. So actually it's a lot of the time that you spend is writing and communicating.
Yeah. You have quite a high social media profile as well. And I guess one of the things that you would do that, as Claire would know, and we've discussed this before, that people will often hold up a study and they'll go, here's one study, look, it shows this, the world now has to change its practice. How do we evaluate studies, and how much weight do we put on individual studies? What's that sort of pyramid of evidence that we work on?
It's really very hard to evaluate a study if you don't really understand the context of the field itself, because usually when you are sort of deep within a field of research, you will know who the main researchers are. They'll be famous. You'll have met them at conferences, and they'll have spoken at all the key meetings, and they will have been publishing consistently year after year and become well established. And what I'm often looking for is studies and papers led by people who I know have been embedded in the field for a long time, working out of well-established institutions and publishing in journals that are highly regarded, peer-reviewed journals. there's a lot of issues with scientific publishing, and frankly, you can find a study to support anything on PubMed. So if you don't understand the field, you don't have that kind of context, it's really, really hard to know if a study is good or not, just from looking at the title, the abstract, and all of that kind of stuff. So there are sort of objective ways of looking, but it's really, really difficult, to be honest, especially when you're looking at the more basic science stuff where it's quite mechanistic and they're using techniques and models that you won't be familiar with. Whereas I think when it comes to clinical research, when we're looking at randomized controlled trials and meta-analyses of randomized controlled trials, that's quite different because actually there are more established benchmarks for how we evaluate those and how we evaluate the efficacy of certain therapeutic agents or approaches, for example. So that's quite different. But when it comes to very basic science mechanistic stuff, it's a lot harder for someone outside of the field to know if that was a good study or not.
That's why it's really important that we have science communicators like Beibei to translate those difficult studies for us, because she's involved in research. She understands how studies are designed. She understands how difficult it is to plan and develop and actually publish the research, that she can give a very, very balanced view on the papers. So, we're lucky to have science communicators that can do that for us.
Yeah, absolutely. And one of the things that you mentioned earlier when we were talking was about the building of a body of evidence, which plays on that sort of meta-analysis there. When you're looking at stuff, how important is that you see, especially in the development of the products that you've been doing, that you see this sort of emerging body of evidence when you're, say, looking for a new ingredient or something?
Yes, well, it's key. We all look to the ingredients that have been around for decades because we understand them, we understand mechanistically how they work, things like retinoids, we know the retinoid receptors, there has been in vitro evidence, in vivo evidence, and then real world clinical experience that we have of these things working. With those kind of things, it's very, very easy to decide on those ingredients. And then there are newer ingredients that are still being researched and are a bit more nuanced and are being used. Things like polyphenols are being used quite a lot in skincare now. Myself and Beibei were just talking about this last night. They're being added to a lot of skincare products, lots of plant extracts. Really, for me, one of the areas that I found most interesting was when I was researching the DNA repair enzymes, particularly because when I first found out about DNA repair enzymes, I think it was probably when Daniel Roche published a paper with Ron Moy back in 2019, and it was really that paper that highlighted to me that these things existed and were out there. And then I went and deep dived into all of the research, a lot of which Dan himself had done. So Dan Roche is basically the pioneer of DNA repair enzymes, and Amanda Rosenthal and Ron Moy and Daniel Roche, basically their published paper in 2019 was kind of a call to action to dermatologists around DNA repair enzymes. They basically framed it in a way where they asked, I think it was like 5 or 6 very pertinent questions. Do DNA repair enzymes work? What is the research? Can they work in skincare? And basically answered each one of those questions. And when I then looked into the DNA repair enzyme story, I was absolutely in awe of these enzymes and how they worked and what their possibilities that they could actually repair DNA in skin. So I think they are a very, very good example of how we need to continue to research ingredients in skincare, and they are not trivial. The DNA repair enzymes were originally researched and studied to be in a pharmaceutical product, so a topical cream that Dan was developing in order to help people with xeroderma pigmentosum. So basically, xeroderma pigmentosum is a genetic disease where your DNA repair mechanisms in your skin don't function as well. And if you had a cream that could be used to help repair some of the damage, then that would be fantastic. So some of his original research was looking at that. And these enzymes, they have been around since the dawn of time, basically in plants and plankton and some animals, some marsupials, but not us. We lost our ability to produce them in our evolution. So there's been a lot of research ongoing, which actually led to, in 2015, the Nobel Prize in Chemistry was won by the scientists who discovered the pathway of how these enzymes work.
Wow.
Yes, yeah, it's called photoreactivation for photolyase particularly. Photoreactivation is phenomenal as a concept, so that's why it won the it won the Nobel Prize. So basically, these enzymes, particularly photolyase. So photolyase finds a little chink in the DNA. So it finds the DNA damage, particularly the cyclobutane pyrimidine dimer. And it sits there and it waits for a beam of light to come along, and it's activated by photons of light to repair that damage, which is amazing. So you can see why, I was deep diving into this. But basically what happened was that, it never became a drug, because to become a drug is quite difficult, to get FDA approval, especially for something that is preventative. And what ended up happening is that a cosmetic company bought the research, and then these DNA repair enzymes kind of got filtered into skincare products. So there are lots of skincare products out there with DNA repair enzymes, and there's some companies who have great products with DNA repair enzymes, but I think they're kind of hiding in plain sight, I would say.
Yeah.
I think there's a few dermatologists now, because there's a couple of us who keep talking about them a lot, like me.
Yeah.
As you can see, I could probably go on all day about this. But I don't know, when did you come across them? Was it just from me and my chatting, or?
I actually came across DNA repair enzymes when I watched a video of an American dermatologist talking about his morning skincare routine, and he was using DNA repair enzymes as part of his morning skincare routine. And I thought, gosh, I didn't realize that this was a thing that dermatologists believed in and used. And obviously I'd come across the concept of defective DNA repair in the context of XP, or xeroderma pigmentosum, but I didn't realize that actually there were things that you could apply topically that could theoretically repair DNA. And I think that there's almost a dogma that we all kind of are taught or believe in dermatology, that most cosmetic skincare can't do that much.
Yeah.
And I think that's built into our training a lot of the time. And over the years, I've realized that that isn't the case. And there's many reasons why we're taught that.
One of the reasons is that
changed,
we are
isn't it?
trained to treat disease. I mean, as
Yeah.
dermatologists, we treat diseased skin, unhealthy skin, and our job is to bring it back to health. And skincare, is really a preventative thing. So I think that's where the difference lies. So a lot of what we're taught is drugs and treatments, and that's
Yeah.
fine. But I think now that we are entering into an age of preventative medicine, that it's really, really important that we focus on these skincare ingredients. And I think us as dermatologists leading that is really important, because we can focus the research, because nobody understands cellular
No.
biology of the skin like dermatologists. I mean, obviously the skin aging scientists in the universities are doing an amazing job, and the cosmetic scientists who formulate the products are amazing. But added into that mix, I really think you need a dermatologist.
For sure, because ultimately we are helping to translate the science and seeing what happens to our patients. And many dermatologists, like Richard Barlow, for example, will be seeing those patients for decades.
Yeah.
And that clinical experience is really, really important
Yeah.
in understanding how topical products can influence skin biology.
Yeah.
And they definitely do. I mean, at the end of the day, when you look at even just a plain moisturizer, it is still altering skin biology. Everything you put on your skin is influencing skin biology. And some things are clearly able to do more than others. To say that all skincare is the same, I think is definitely not true. But I think that there is a bit of a knowledge gap in our training.
Yeah.
Sort of our default training anyway.
It's interesting, because one of the reasons Richard got involved in this was because of that, because of just from years of seeing his patients and the difference with the patients that had actually used topical skincare. And obviously myself and Richard, in our practice, we deal with a lot of skin cancer. Being Mohs surgeons, we tend to be referred a lot of skin cancer, kind of photo damage, precancerous lesions, and our specific patient cohort, a lot of them might live abroad for 6 months of the year. So a lot of them are quite photo damaged. But definitely, when I started working with Richard, he said, this is definitely a thing. People who have a good topical skincare routine just do, and it's more than SPF. I think that's what we're learning now. Obviously, SPF is the pinnacle of, you definitely need to be applying SPF if you want your skin to last, your lifespan. But there are other active ingredients now that we know that work.
Yeah, it certainly has been a revolution. Coming back to what damages our skin in the first place, because prevention is always better than cure. The World Congress on Melanoma has just been held in Brisbane, and they basically said that the global rates of melanoma are growing staggeringly, and in the UK they are up year on year by about 7%, and even in Australia, where we've had slip, slop, slap messages for decades since the 70s, they're up about 3% every year. So clearly we have something that is damaging our skin skin going on there. So what is it that the sun is doing to our skin?
And we always talk about melanoma rates, but obviously there are other skin cancers. The keratinocyte skin cancers, basal cell carcinomas and squamous cell carcinomas are far, far more prevalent. And while melanoma is definitely more lethal, there are now a lot more squamous cell carcinomas, and that means that there are a lot more high-risk squamous cell carcinomas, which unfortunately also can be fatal if they are very aggressive. So we need to talk about all the different types of skin cancer, but your two main risks for developing skin cancer really are your cumulative UV exposure and your age. Things like melanoma, definitely the amount of sunburns in your lifetime and the number of moles, and your genetics definitely play a part. But you can't alter your genetics so much, but you can alter your UV exposure. And if you're living longer, you can try and reduce that cumulative UV exposure that you get over your lifetime. You know, we don't want people to stay inside and avoid the sun. We absolutely see the benefit of going outside, enjoying the outdoors. Baby is very, very sporty. She does a lot of outdoor sports. It's really important that we get out there, and that in turn increases your longevity, but you need your skin span to match your lifespan. You know, skingevity has this new term. But we're not saying completely avoid the sun. We're just saying that take measures, be sensible. If you're sitting outside, try and find the shade, wear a hat, put your sunscreen on when you can. Every one of us is going to miss out and forget it sometimes, but it's just that cumulative UV exposure. If you can get your curve down a little bit, that is going to really impact your skin in the long term.
What's the sun actually doing to our skin at that level under that we can't see?
So it's the ultraviolet rays that are very highly energized that are bad for our skin cells. So UVA and UVB in particular. And UVB, which causes burning, basically directly damages the DNA in your skin cells. So it immediately causes damage. The UVA, basically, it penetrates a bit deeper into the dermis, and it can cause things like reactive oxygen species. So that if you've heard of free radicals, they basically are cells that are very, very energized and are stealing electrons from other cells around them. And in doing that, they're damaging all of the other cells. They're causing a lot of inflammation. That in turn kind of spirals into more damage and cellular damage and structural damage. And then the DNA mutations build up. So you've got a combination of what we call genomic instability, and then you've got inflammation, and then with that your repair processes as you age don't work as well. So when you're younger, you have repair processes like your DNA repair, nucleotide excision repair, which are all primed and ready to repair things. But as we age, everything starts to wane a little bit. So if your immune system isn't functioning as well, and you know our skins are a very important immune organ, and the immune system in your skin starts to wane as well as you age, and you get a lot of inflammation, you get a lot of genomic instability and DNA mutations. That's when things all start to tipple onto the road of precancer and then possibly the development of skin cancer. I mean, precancerous lesions in themselves are something that we don't want. Myself and Beibei work in the NHS, and we see a huge, I would say, burden of disease for precancerous lesions. Even yourself, you're doing your Efudex today, Fiona. You showed us earlier on, which is great, you know, but you're aware of that. But that is precancerous disease. So it's all around us. Something like 1 in 4 people will get an AK in their lifetime. So,
I'm a walking
it's really, really
keratosis.
prevalent. I mean, we're not trying to frighten people, but we're just trying to say that this is something that happens. It's mostly because of UV, it's partly because of aging, and that's why we're investigating aging too.
In terms of aging, I mean, it's not, we hear a lot about collagen and, you know, being the sort of scaffolding in the skin that sort of gives it its firmness. But what else does the sun damage under the skin?
Aside from collagen, that's one structural protein, but there's lots of different structural proteins within the skin. And it's interesting what you say about DNA damage, because I think actually probably when it comes to things like squamous cell carcinoma, basal cell carcinoma, a lot of the research that I've been involved in has actually looked at some of these cancerous changes, and actually it's quite multifactorial. So we know that things like fibroblasts, which are the cells that make collagen, are also involved in the development of skin cancers. And if you think about it, when we're exposed to UV light, you have the UVB damaging your DNA, and actually there's research that shows that in completely normal skin of a 30-year-old in a sun-exposed site, there's thousands of DNA mutations already. So completely clinically normal skin has all these UV mutation signatures. So we are just building them up and building them up. And at some point, something tips where these become precancerous or develop the potential to become precancerous. And there's a lot of emerging research about how this is actually an interaction of these cells which contain these mutational burdens, and also supporting cells such as the fibroblasts and things like, we know that if you have chronic scarring, that is a risk factor for skin cancer as well. Chronic inflammation and disordered protein in the skin, in the stroma.
And disordered architecture.
Exactly, disordered architecture. These create permissive environments for these cells to become unlocked and form cancerous lesions.
Yeah. There's a finite balance in our skin all the time. As a seminal paper where they looked at the blepharoplasty skin, so from eyelids, so when people went to have their blepharoplasties done as a cosmetic procedure, the skin was examined. And something like a quarter of the cells already had UV signature mutations. So we have all of these DNA mutations in your skin, but your immune system is basically kind of working and keeping them at bay and nothing is happening. And it's when the balance tips that you can develop precancerous change and other things. We're talking about one end of the spectrum where there's skin cancers and precancerous lesions, but there's also photo damage, pigmentation, wrinkling, all these other things that people don't want to get, and they're all kind of on this downstream trajectory.
It's almost like a spectrum where you have healthy, youthful skin that's not sun exposed or minimally sun exposed, and then as you accumulate UV damage, that damage obviously will influence our keratinocytes, and that will manifest as some sort of skin aging with the DNA mutations. And then over time, those mutations and also the damage to the dermis and the collagen structures and the fibroblasts, all of those create an overall environment which is much more prone to skin cancer and more obvious skin aging. With any kind of skin condition, there's always kind of invisible stages of the condition occurring, and that's something that we see across different skin conditions as well, like eczema as well.
Yeah. You mentioned photoaging there, and that's one thing that people obviously worry about as well. Well, except for people like me who still are recidivously sitting in the sun. But other people who have half a brain do worry about photoaging. And collagen is one thing, and fibroblasts. But what about fibrillin and other things like that? What's their role?
And you've heard of collagen. Everyone's heard of collagen, whether it's in the skin or collagen supplements or collagen banking. It's such a buzzword. Everyone loves talking about collagen these days. A lot of the research that was done by the team in the University of Manchester, who have some of the top skin aging scientists in the world, looked at fibrillin. So fibrillin is a microfiber that is located between the epidermis and the dermis, and these little fibrillin microfibers kind of act like little hooks between the dermis and the epidermis. They have a kind of they describe it as a candelabra formation, and they are there giving structural support to the epidermis. But what's interesting about them, and a lot of the research that they've done looking back over years has looked at, well, are they a marker of skin aging and are they a marker of photo damage in skin? But they're a very good marker of both, but particularly photo damage. And they actually they compared unphoto exposed buttock skin of younger people and of older people to see, and actually the structural changes were very similar. They both had good structure and structural support, and obviously the aging skin, which is intrinsically aged, had some changes, but not a huge amount. But then when they compared it to the photo age skin, there's a huge difference, and one of the major differences is this loss of these fibrillin microfibrils. So it's a really good marker of photo damage in the skin. And what's interesting about that, that in itself is really interesting, because that obviously leads to wrinkling and sagging and loss of elasticity and things like that. But what's interesting is that you can monitor how it is reversed in studies. So basically the Manchester patch test assay is a way of looking at how fibrillin fibres can be restored. So the fibrillin microfibrils between the dermis and the epidermis are diminished in photo-age skin. And the interesting thing about them is that you can not only see that they have got diminished in photo-age skin, I mean, you can see a gap called a Gren zone, but you can also see when they are restored. And there are certain things that can restore fibrillin microfibrils, and particularly topical skincare. And for many years in the University of Manchester, they studied things like tretinoin and showed how good it was at restoring fibrillin microfibrils, as well as collagen. And they looked even at things like the retinols, which are in over-the-counter skincare products, and they looked at them at different levels, because one of the problems with tretinoin is the irritation. So it was always very interesting to see, well, actually, do retinols work? And aldehydes and retinol esters and all those things. So
peptides.
Yes, and peptides. So they're looking at all of those things and seeing whether they worked on the fibrillin microfibrils, and they did. Through years of looking at these things, they then figured out that they could recapitulate the restoration of these fibres by doing an accelerated test on the forearm of real humans using little patch tests of creams, and then looking at whether the fibrillin microfibrils were present or not present. So basically that was a good indicator of whether a cream was going to work or not. And that way of studying skin aging products is called the Manchester patch test assay, and they've been using it for years in lots of studies in the University of Manchester and now around the world. And it's kind of seen as one of the gold standard ways of looking at different products, used in the university as an academic test, but is also used sometimes by industry, and sometimes it's just done to look at surface level changes, and then sometimes they actually biopsy the skin and look at the skin under the microscope. But that in itself requires a lot of ethical approval and planning to be able to do that. But basically the test itself requires real human skin, and
human participants,
and
not just the skin.
Yes,
Not just the
true.
skin.
Not just the skin.
Because a lot of research, they would use skin explants, so people who have donated tissue from some sort of plastic surgery like abdominoplasty or mammoplasty. So they are using skin, but skin that's been removed from the human is still different to living human skin.
Yeah.
Because obviously when the skin is on the human, you have circulation, you have the immune cells communicating from the skin to your central immune system and so on. So
also,
it's completely different.
A really important thing about that study is the fact that each person acts as their own control.
Yeah.
So when you're thinking about how a study is powered or how many people are in it, for a histological study like that, having an area of skin that is its own control actually is great, because it really validates the research.
The Manchester patch test assay is an in vivo accelerated cosmetic testing protocol, and in vivo means that it's done in a living human. So the skin is influenced by what's going on in that person's body, how their immune system is functioning, and all of the other factors. So it's obviously much more indicative of what might happen if someone uses a product in real life compared to something that's being tested on skin that's been in a dish donated from an abdominoplasty, for example, which is something that we commonly use in research. And we know that putting things under occlusion is a way of accelerating its power, because it essentially forces the penetration of the product at a much higher concentration than it otherwise would be. So we use that a lot in dermatology anyway. So if we want something to work a bit faster, we basically put the topical prescription cream on, and then we cover it in a hydrocolloid dressing. And that helps us achieve greater potency with whatever we're using. And they applied this principle when they developed the Manchester patch test assay, because it meant that rather than having to recruit a group of people to use something for months on end, they basically were able to benchmark, okay, if you do it under occlusion for a shorter period of time, we know that this is about the same as 12 weeks worth of daily application. Because a lot of the problems with research studies that go on for long periods of time is adherence can be an issue. People might drop out of a study. When you're doing a study, they have to adhere to very strict criteria a lot of the time in terms of what else they're doing. You run into all kinds of issues. So this accelerated in vivo study actually provides a very useful model for understanding how a skincare agent may influence skin biology in a way that's a lot more feasible and practical, and therefore useful to cosmetic companies and also just for skin biologists who want to understand how something influences the skin.
Some people might say, well, the skin on your arm isn't the same as your skin on your face, so how valuable is the result?
It isn't the same. I've been involved in research analysing how the skin on the face differs from the skin in the body, and it is definitely different. So key things that we identified were the vascularity is very different. I mean, you would know that as a skin surgeon anyway, but the face is a lot more vascular. It's a lot more rich in pilosebaceous units, which is like my favourite part of the skin biology. And that does alter the skin biology a lot. But when it comes to examining photo damage, the arm is a very acceptable surrogate for facial skin, because if you think about it, how many people are going to be happy to donate facial skin biopsies? It can be quite challenging.
People's forearms are usually actually quite photo damaged. My forearm is definitely more photo damaged than my face, because I'm much more diligent about putting my creams on my face. So it's a good marker for photo damage. And if you're looking at things like fibrillin particularly and collagen, well then it's a good marker for that. And I suppose when you're thinking about the Manchester patch test assay in particular, you're comparing forearm skin to forearm skin. So you're not comparing facial skin to forearm skin, if you know what I mean. So you have your control there. So all you're looking for is those changes. Did it work? Is there more collagen? Is there more fibrillin? If there is, can you quantify that? And that is one of the things that they do as well. So they can quantify both the quality and the quantity of the collagen in particular. And also one of the things Abigail was telling us in Manchester was that the quality of the fibrillin microfibrils themselves is very important, because you can have regeneration of these things, but sometimes the regeneration might not actually be functional. You might just have more of something, but if it's not working the way it should. So she said it's really interesting that they can actually see that the fibrillin microfibrils are restored to that candelabra structure, which they know is how it functions in the skin.
If the skin on the face is more vascular than the forearm, then would you actually expect, once you put that on the face, that you'd actually get a better result, because you've got skin with more vascular, more nutrients, more...
It would be a very interesting study to do, I think, because you would do the Manchester patch test assay on the forearm of that person and on their face at the same time. You might not get ethical approval for that, though. And that's one of the biggest difficulties in a study is getting ethical approval.
But also just getting patients to, or participants to consent.
Yes, because you're causing a scar, so people don't really want a scar on their face.
So most research studies that have looked at cosmetics, the vast majority have done a combination of non-invasive testing with some sort of photography tool, maybe with some sort of supportive mechanistic work. So the Manchester patch test assay is a bit of a hybrid study where it combines some a bit of clinical data with mechanistic data, and I think that's why it's quite a unique approach to take.
Very unique, because as well it's final formulation testing. The testing that goes on individual ingredients, which is really, really important. A lot of testing now in vitro is done on peptides, and we know niacinamide has this amazing action on mitochondrial DNA. But this is final formulation testing, because it's very easy to kind of say, well, these ingredients look like they work in a petri dish, but actually do they work in real human living subjects? That's very, very different. So it's really actually definitive. It's very difficult to argue against when you physically see something under the microscope, and we can all kind of just look at the images, even if you don't want to read the study, and you can see there's just more collagen and the fibrillin has been restored. So there is an obvious architectural change there.
What I find interesting about the fibrillin story, and this is aside from the science, is that people are always asking about crepey skin. That's a really, really common question. And if you were to look histologically at crepey skin, it's probably skin which has that Gren zone.
Yeah, absolutely.
And without those fibrillin microfibrils, because crepey skin often has that very light, fragile texture to it, with very, very, very fine wrinkling. And I think that's that loss of integrity between the epidermis and the dermis. So I think probably fibrillin is very important in that kind of appearance.
We need to talk more about fibrillin and less about collagen, although collagen is important, and more about DNA repair enzymes.
You mentioned earlier, just to take you on a completely different area for a second, you mentioned inflammaging.
Okay.
So what is inflammaging, and why does that matter to our skin?
So inflammaging is obviously a very broad term, but we do know that in general, levels of inflammation, so that's a very broad term, we know that as people age, there is a shift in the immune composition in the skin. So the aging skin phenotype is clearly very different in a number of ways, but the immune system is one of those ways in which it ages. So we know that the cytokines and the immune cells are different. For example, pruritus is a very common feature of skin aging, and that's something that we see a lot in our clinics.
What is pruritus?
Itch. So chronic itch, chronic unexplained itch is very, very common in the elderly. People often complain that their skin is getting more and more dry.
Yes.
And the first place we tend to see this is the lower legs. So even if you've never needed to use cream, at some point in your life, you might have to start putting cream on your lower legs. So that's, I would say, is one of the early.
There's a kind of a chronic low-grade inflammation that just kind of creeps up as you get older. And it's not just in your skin, it's in all of your internal organs. And that's a lot of where a lot of the anti-aging research is happening now is in oxidation, which is one of the major factors in inflammation, reactive oxygen species. In the skin, we see matrix metalloproteinases are a major factor. And anything, we talk about the exposome now, and people often ask what the exposome is, but it's all these environmental factors that you're exposed through all your life. So, UV, pollution, lack of sleep, stress, all of these things we know increase your levels of low-grade inflammation, and that in the long run is aging.
Preventing things. This is one of the, sunscreen is one of the great preventers, as we know. If you're looking for a sunscreen, what are the things that the average person should be looking for when they're cruising those supermarket shelves?
From my point of view, broad spectrum, something that you like and will actually use. I think there's a big difference. I like to separate it into, your day-to-day sunscreen, basically, that you're going to, use all the time, and then the thing that you're going to use when you go on holiday. For me, I treat them quite differently.
Me
so
too.
So I think there is a difference, and I think sometimes that might be what we don't get across to people. When I say to people, you should be putting sunscreen on every day, some of my patients look at me like, what, my Hawaiian tropic or whatever. For something that you're wearing day to day, if it's broad spectrum, anything over SPF 30, and broad spectrum means that it's both UVA and UVB, and something that you enjoy using. After that, it comes down to whether it irritates you or not. And, if you've sensitive skin, you might want to go with a mineral sunscreen. If you've pigmentation, you might want to go with something with a tint in it. So there's nuances, but the basics are something that you like, that is broad spectrum, that is SPF 30 and above. Just to make it simple for people. Then you're going on your holidays. Just because you're going to probably be spending more time outside, I would definitely go with something that's a higher SPF. The formulation doesn't have to be as nice because you're putting it on your body rather than on your face. So anything with a good high factor and a good UVA protection. The sunscreens now that we're kind of talking about are what we call active photoprotection. So you're going to hear a lot more of that, because the SPF, the UVA and the UVB filters basically are just, they're blocking the UV rays. But active photoprotection is where you start adding things like antioxidants and DNA repair enzymes and things like that will actually help prevent and repair some of the damage as well. So that's kind of where the industry is really focusing now, I think.
Yeah. And I'm similar to you. So I very much have my everyday, day-to-day sunscreens, and then I have I'm going into a high-risk exposure environment, performance sunscreens. And the things which I use for performance sunscreens, I look for water resistance, apply to wet or dry skin.
Well, do they have those?
Yeah.
To even wet skin as well?
You have wet skin specific sunscreens. You have sunscreens which have been tested for being water resistant for 8 hours. There are all kinds of additional things. So the problem is I think the least effective sunscreen is the one that sits on your bathroom shelf and doesn't go on your face. So I think the number one thing for daily preventative action is the most cosmetically pleasing, enjoyable sunscreen to use. And actually any sunscreen is fine for that. But obviously some may be more cosmetically pleasing or enjoyable than others. And that's kind of how I generally frame it. And then you have all of these other added extras in your routine. It's such a nuanced discussion, because a lot of the time you're being asked these questions by someone who may be spending a lot of money on other skincare products. And really there is so much in terms of investment, like investing in a nice sunscreen is actually very, very worthwhile.
It's a difficult one, this, because I remember saying to you a while back, I can't believe how expensive these things are. But you explained, basically, when I was growing up, back when the dinosaurs roamed, literally all we had was white zinc. And no self-respecting teenager was going to walk out with white zinc anywhere on their face, and you certainly couldn't put it on your body. So your entire body was exposed, and if you were a teenage girl, you just weren't wearing that. So there was literally pretty much nothing, but it has massively evolved, but it is incredibly expensive. And part of this, I think, is a tax as well. If you're looking at 30 quid for, a little thing like this to go on your face, is there an argument for saying that this is preventative health care and the tax should be removed?
Well, I'm going to be in Parliament tomorrow, I think, to discuss that. There's a UV safety and UV inquiry launch. There's lots of nuances to that, because if you do drop the tax, you don't want companies just reaping the benefits of that. You would have to have a way to know that that is actually going to the customers. Part of me thinks that maybe you should keep the tax on it, but use all of those tax funds for education and also for, I think we should have sunscreens in schools. I think you should have pumps in schools so that kids just get used to it and they're told, before they go out that they use their sunscreen. And those kind of initiatives I don't think would be that costly. I'm sure dermatologists would happily give their time to educate people. I know there's been some surveys done that have shown that people aren't as educated about sunscreen as we think they are. So part of me thinks that rather than just dropping the tax is that maybe you relocate the funds to be used specifically for that purpose. I know in the US and in the
skin sun clinics that you can go to, because you can't get a decent skin check in this country. I'm sorry, you just can't get a decent skin check in this country.
Ouch.
Ouch.
And the thing about the filters is, because you bring it out a good point, so the kind of high performance ones that Beibei is talking about, really still probably do have some element of that zinc in them. The ones that will stay on your skin and last all day. I mean, there's this amazing one that one of my patients got me from Brazil called Brazinco that I put on my daughter now, and it's basically skin coloured, but it's completely impermeable. I mean, it's impossible to get off, which is great if you have kids that are in and out of water, and she's very, very white. But the filters are basically, that's where the science has developed. The filters in sunscreen now have really, really come on, and there's some really fantastic filters. Some companies have made their own, which are great. It basically means that they are much more cosmetically acceptable and that they are much more photostable and that they work better. So in our daily defense, we have these encapsulated SPFs, which are really amazing because the formulator absolutely loved working with them and was very, very excited to get to work with them, because he could create a day cream that was really cosmetically elegant without having to worry about the levels. You have to put a high level of UV filter actually into a cosmetic formula to get up to that IUVA+++, the 4 pluses and the SPF 50, and that is sometimes what gives it that texture that people don't like if it's too oily or too chalky. But with encapsulated filters, that gets rid of all of that problem. The filters are basically in these little bubbles, and they are of a size that they cannot penetrate the skin. So because they're 1 micron and they're basically double encapsulated, they can't systemically absorb, and that means that they don't irritate your skin as much as some chemical filters can. It means that they're really photostable, and it means that they can be put in really elegant formulations. It also means that they're very clever in that they are negatively charged, and by doing that it means that they're even less likely to cause problems on your skin by absorbing, but it also means that they just form this totally even film on your skin, so there's no holes that sometimes can be a problem with other filters.
Because the other ones sort of tend to clump together, yeah?
They can, it's called flocculation, yeah. They can clump together. Some of the nano zinc or nano titanium dioxide mineral filters can sometimes flocculate, on the skin.
Now, you mentioned there that it's not absorbing, because there's one thing that that I think comes to you in many ways, is that some people get very worried about the ingredients in skincare products, especially sun care, and they've got their little apps and they go through and they look at them and they look at the environmental working group and virtually everything is toxic and it's going to kill you.
You wouldn't go out the door if you read all that and
no,
you
you
worried
wouldn't.
about everything. I mean,
yes, what people fail to realise is the quantity that, if you're going to put yourself into a vat of stuff, then it's vastly different to having something in a very low dose. Even if they are using a sunscreen that does absorb, should they be worried about that?
No, because at the end of the day, cosmetic ingredients are heavily regulated.
Yeah.
I'm not a toxicologist, but I know that in the UK and EU and across the world, there are scientists who are specialists in evaluating hazards and risks and what dose might pose a risk to human health. And the problem with a lot of these ingredient rating apps is that they don't take into account how much of an ingredient there is and how it's being delivered. So a lot of these studies that they're quoting are completely taken out of context and not actually applicable to humans. Yeah. And it's a very controversial area. And I think a lot of the time this is something that comes to the fore when women are pregnant and they start looking into what they're eating, what they're putting on their skin and all of that kind of stuff. And as soon as you're pregnant, you're kind of bombarded with a lot of this information. And I think it's a very vulnerable time and people get sucked into all of this stuff. And I can appreciate why it could seem concerning, but at the end of the day, you kind of have to trust the science
experts.
And the experts. And
Yeah,
those are the
toxicologists
yeah.
spend their lives researching this stuff, the dose and the risk, as Beibei said. So when it comes to the regulation, you're not just relying on one toxicologist and their opinion.
And one study.
And one study. You're relying on a consensus of experts. And I mean, I think that is, as dermatologists, we rely on them. If the toxicologist turned around someday and told us that there was a problem with something, we would perk up and listen. But when they're telling us this is not a problem at this dose and these levels, and they have tested them to the nth degree. I mean, when you formulate a cosmetic product, you get essays and essays of reams of information about every single ingredient and all of the testing that has been underdone on those ingredients. And there are EU regulations and regulations in the UK. Interestingly, the cosmetic formulations, I would say, are much more highly regulated than in the aesthetics world.
The other thing that's important out of those sorts of things is we come to the end of summer, we've got our nice big batch of sunscreen that goes back into the cupboard. Summer rolls around again. Is it all right for us to take that one that we had last year, or has that period of time between last summer and this summer affected the stability and the efficacy of those products?
Probably yes, because every sunscreen, every product is different, but there is a little label that you'll find on every product which says how long it's good for after it's been opened. And that's because the stability of that product cannot be guaranteed after that period of time, so the formula might start splitting. And that's something that people sometimes see if something's been left out, and especially if you're keeping it in your car where it might become overheated, it might accelerate the formula kind of going bad and not working as well. So one thing that you touched upon with the flocculation is that when you apply a sunscreen, you want it to form what's called a sunscreen film on your face. So you may have heard you should apply a sunscreen and wait like 10 minutes before it's like activated and then you can go out in the sun. And a lot of people think that it has to somehow react with your skin to be activated. It's not really the filters activating, it's actually the film forming. And that once that film is formed, that's how the sunscreen filters are able to filter out the UV rays. So it's this film formation process. And the encapsulation really helps with that film forming process, essentially.
Is that the same for mineral ones as well, or is it only the chemical ones?
It is, and it's quite a common myth that mineral sunscreens instantly work because technically they still need to form a sunscreen film as well. So you can basically, when you apply sunscreen, there's always this kind of dry down period where it might look slightly shiny and feel a bit tacky on your skin, and you have to sit and wait for that to pass, allow the film to form, and then apply some makeup or whatever over it. Actually, I've even seen some cosmetic chemists argue that sunscreen should be the last thing you put on your face, so that the film forming is optimized for protection. But I know usually people do sunscreen first, then makeup. But you should theoretically wait some time between the sunscreen application and the makeup because you want that film forming to take place first.
Yeah, it's a tricky one, that one, isn't it? I mean, I don't wear much makeup, but I could imagine that if you had a quite greasy, heavy sunscreen, you couldn't put makeup on over the top of it. But then again, vice versa, you probably couldn't put it on top.
This is why I like my patients over the years, when I say to them, you should be putting on some sunscreen, have always said, well, it's just an extra step. So that's why I like if your daily sunscreen doubles up as a moisturiser, because then it's not that extra step. I like to put on a serum and then my moisturising sunscreen and then my makeup. That's basically my three things. But if you don't even have that time, or like you don't wear makeup, I mean, you could just put on your moisturising sunscreen, and then that we would be happy with, because remember, the sunscreen's the most important thing out of everything. So I think if it doubles up as a moisturiser, usually, especially women of a certain age, we all want a bit of moisture in the morning. So most people are actually at least putting on a moisturiser. When we were developing age defence, that was a key part of the development process, that we wanted this formulation to not just be a really great sunscreen, but also to be a really great moisturiser. Basically, I developed it for me. I was thinking, what do I want? And this was exactly what I wanted. I just developed the perfect product that I know I would like. So
excellent.
Hopefully other people do too.
I think that's a great R&D model.
Yeah. What is? But I think that there is that with skincare, especially if you're a dermatologist and you see all these great products coming out. Yeah. But sometimes you do think, I wish somebody would make this. So it's nice to be in that position.
It's got all
now.
your favourite things in it. It's got SPF filters that perform well because of the encapsulation technology. It's got the DNA repair enzymes and it's got antioxidants.
Yes, the polyphenols.
Yeah.
Which I like.
Yes. Now, going to the polyphenols, you touched briefly before about basically fruit and vegetables in our skincare.
I know, yeah.
So what can we actually learn from these plants that actually makes our
this is
skin
the
better?
research that is kind of nuanced and really just developing. So we're beginning to realise that polyphenols, which are basically molecules in plants, are packed with things like flavonoids and different molecules, things like quercetin and rutin and tannins and all of these things that we actually realise are very, very good for our skin. And in skin ageing science, so Professor James Kirkland, who's a skin ageing researcher in the Mayo Clinic, is coming to talk to us in the BAD meeting this year. He researches mainly quercetin and fisetin. And these are two plant flavonoids, basically. I mean, he uses them at much, much higher levels. So quercetin and fisetin, you can get them in like strawberries and apples.
And pears,
but you'd need to eat a lot of strawberries and apples to use the doses that he uses. But, so he's doing these experiments. They're called senolytics. So senotherapeutics are basically drugs that are either senomorphic, so they revert the aged senescent cells back to being normal, healthy young cells, or they're senolytic, and senolytics basically get rid of the abnormal senescent cells.
Sort of scavengers.
Yes. So in skincare, you will see a lot of the bigger cosmetic houses now will put a plant extract in with pretty much everything that they're doing. So, Eucerin did some amazing DNA methylation studies on pine tree extract, and Heliocare have added some rooibos into one of their products now. They're famous for using Polypodium leucotomas, which is a fern, which is known to be a very good flavonoid. So, we're learning a lot more about these individual chemicals that happen to be in plants and how they are actually, so with the bilberry that we use, it has a very high level of quercetin, so especially when it's actually made into its extract form. So bilberry extract, when it's in its kind of powdered form, has about 50% quercetin. It's known to be very anti-inflammatory. It's been shown to reduce kind of MMPs, neuroreactive oxygen species, and in the Manchester patch test assay, in the test of the night repair therapy, the combination of the plant polyphenol with the other ingredients had a kind of a synergistic effect. So there's still a lot of research that needs to be done in this area, but it's really, really fascinating, and it's probably one of the ways that science is going. And that's why the skin aging science is so interesting, because with skin, we are very lucky as dermatologists because the skin is so accessible. If you think, well, skin aging is kind of cosmetic, it's not really, because aging itself is not a disease, but it is one of the biggest risk factors for all of the diseases that we see. Understanding aging is actually really important for us to be able to manage age-related diseases as we go forward, and things like understanding why these polyphenols work, what are flavonoids doing in our skin. Once we understand that, then we will be able to help with the aging research for the rest of your organs, basically.
There'd be some that would argue, well, hang on a second, there's just not enough evidence behind these at the moment, so why would you bother putting them in?
I think the evidence is quite robust now, actually. There may not be one amazing study, but as we were talking about research earlier on, there are lots and lots of different very, very good studies. And when you look at the body of evidence, and when you look at the scientific consensus, and then when you see that there are some major researchers like those in the Mayo Clinic who research aging, are using these same compounds, then it all starts to add up to a story that makes sense.
And you're also looking across types of research as well. So when you think about the whole antioxidant story and how that works, what is the effect of antioxidants on the human body? When you look at what reduces your risk of things like heart disease and cancer, we know that people who have an antioxidant-rich diet, i.e. eating your 5 a day, it doesn't get more simple than that. That is a health recommendation that's been around for decades across pretty much every country in the world, to eat your fruit and veg. And fruits and vegetables are your main sources of antioxidants. And we know that people who eat more antioxidants also have reduced skin aging as well. There's been epidemiological studies where they've tracked people over time, and that's been an observation that's made. So antioxidants are good for your general health, and therefore good for organismal aging and good for skin aging. And then when you look at topical antioxidants, there is a lot of mechanistic data supporting the importance of topical antioxidants. And obviously a lot of this was first pioneered by Sheldon Pennell, looking at vitamin C, and vitamin C is the most abundant cutaneous antioxidant, but there are also other antioxidants as well within the skin that are either synthesized or obtained through the diet. And the skin is obviously an amazingly accessible organ. And the amazing thing is we know that we can apply topical products to the skin and influence the skin biology. We cannot do the same thing with as much ease to our other organs. We kind of have to modify our diet to try and improve the long-term health of other organs, which is a lot more challenging because there's a lot more factors at play there. Whereas with skin, we have the ability to apply something topically and influence skin biology. And I think the antioxidant story, many years ago, people maybe were less convinced, but over time, there's just been so many different studies. Not all of them are the best studies, but when you look at the body of evidence and also the variety of antioxidants that have been studied, it's enormous. So many different antioxidants have been shown to be efficacious.
The dietary aspect and longevity, that is one of the reasons why the tomato skin was studied, because the Mediterranean diet, everyone knows that has major health benefits.
Does it concern you then, though, when people start saying, well, hang on a second, I mean, I saw this the other day, a woman basically saying that she couldn't eat strawberries because of the sugar in them. And, you know, we have this whole thing at the moment, the glycation and this sort of inflammaging and things like that. So does it worry then when people start saying, but I can't have all of these things in my diet because
I think it's not about just eating one thing. It's about eating a rainbow of fruit and vegetables, and we know that that's where all the benefits come from. And that's why I think the plant polyphenols and plant extracts are interesting, because they are not just one chemical compound. They are a synergy of different things. But reducing sugar in your diet makes sense, but refined sugars makes more sense than reducing things like fruit.
You mentioned there's synergy. So when we've got these antioxidants or these extracts, we know that, you know, vitamin C, ferulic acid and vitamin E work quite well together in skin. So is there an argument that having a nice mix of these things in a product is better for the skin?
Vitamin E, again, is one of the, C and E are basically the most prevalent antioxidants. Vitamin E is a very good backup antioxidant. It kind of supports other antioxidants, so it's actually a good one to put in with the plant extracts. It can help stop them from oxidizing themselves because it can donate electrons. So you will often see vitamin E actually in skincare products as one of the commonest ingredients in skincare products because of that. But they seem to have this kind of entourage synergy effect, and they all work in different areas as well. So some of them work in the kind of lipid-rich areas of the skin, some of them work in the more water-rich areas of the skin, and that's why using different ones, and it makes sense. There was always the question as to whether antioxidants with your sunscreen, if they were doing anything beneficial, and there have been a lot of studies looking at SPF with antioxidant or an SPF without antioxidant. I think the problem with that has been that with the SPF filters, they can interact in the formulation, and that is something that we need to study more. But again, with things that are encapsulated, that really helps, because you're keeping those ingredients from reacting with each other, and they're encapsulated and developed in a way that they will only act in the areas that they're supposed to act, and that stops them from having those kind of interactions in the formulation.
Yeah. You mentioned before the DNA repair enzymes. Are there other areas as well that, when looking ahead, that are interesting that you'd like to see formulated into skincare? The sort of mitochondrial RNA or other sorts of gene snipping type technology and stuff that's going to actually improve our... Could they be a reality in skincare?
I think the thing that we want to investigate is things like peptides. So there are different types of peptides. There's cell-communicating peptides, matrikines, and some of them are kind of collagen peptides or collagen amino acids, and they each have different functions in different areas. And one of the things that Professor Mike Sherratt is studying at the moment is where these different peptides work. There are lots of peptides and they all do something interesting. So I think that's a huge area of research. And then how these ingredients are actually absorbed across the stratum corneum, where they are going and what they're doing. I mean, there's huge areas that we still need to study on all of these things.
I know that from a research perspective, there have been developments in things like topical gene therapy to help patients who have specific genetic diseases, for example. The logistics and the research and the infrastructure behind developing this kind of a product is quite complicated, and it was probably more likely to be in a pharmaceutical agent than in a topical cosmetic, although these things often turn out to be surprising. So exosomes, for example. Exosomes are these really, really tiny molecules that are secreted by cells, essentially, and they are little packages of messages that are secreted by cells, and depending on the cell of origin, that package might be slightly different. And a lot of exosome research has been done because people think that if we can capture these natural messages that are being sent by certain cell types, then we might be able to accelerate wound healing or promote stemness or stem cells and things like that. Unfortunately, the term exosome is thrown around because people have this vague association of exosomes with stem cells, and they think that all exosomes are basically stem cells, but that's not true at all. It's almost like a delivery system. It's a biological delivery system, and then the package being delivered is dependent on the cell type. But what I find interesting is that exosomes probably are a really important area of regenerative medicine and regenerative dermatology.
And medicine.
And medicine.
And drug delivery.
Exactly. But because it is so challenging to get things through the sort of pharmaceutical route, it's been sort of co-opted in the sort of commercial space. And you have exosome science that is probably not 100% there, but halfway there, that is being used in a lot of topical skincare. And the regulation and the science behind it is not the same as if it was a pharmaceutical kind of product. And I think what you end up with is you have a lot of ingredients that fall into a grey zone from a regulatory perspective. And although I welcome all research and any kind of innovation, I think that it would be a shame if exosomes never fulfilled their medical potential, because what we want is to actually help our patients who do have a real problem with skin regeneration or have skin failure of some sort that we are seeing as emergencies in the hospital. It'd be great if we could offer some treatment for that, but you know, a lot of that.
It is fascinating. There's a Society for Extracellular Vesicles, which are the real scientists who do a lot of study in these things. So that's what they are. They're like the secretions of the cells that control the other cells around them. And as Beibei said, you know, the best ones come from basically umbilical cord or something like that. Very young, youthful cells that secrete these. The secretome now is kind of the broader term for all of the secretions from the cell. But there's no standardization of that process for cosmetics. These cells, these little organelles are actually living. So their survival in whatever medium that you're putting them in, that's dubious. You know, is anyone testing them? You know, we know that they have to be stored in a certain way if they're going to survive. And their contents are actually really, really important, because you don't want to be putting little messengers on your skin that are going to cause inflammation. You want to be putting messengers on your skin that are going to, you know, help your skin and repair. But the science is really fascinating, and there are a lot of researchers, I know particularly actually in Singapore, who are doing a lot of really interesting research in secretomes for wound repair, and exosomes could be kind of programmed to go to different areas within your body to help with disease. So I know there are research groups who are studying putting different peptides, which are like messenger peptides, that will tell the exosomes where to go and which cells to interact with, and then deliver their load basically. So it's all really fascinating, but it's in its infancy, I would say, the science. So we try not to get too carried away with the flashy kind of new terminology, and just dig into the science. And sometimes actually when things become so popularized, that becomes quite difficult. It becomes difficult to see the woods from the trees. So again, you know, that's where we need kind of real science communicators like Beibei to kind of wave the flag for the real research that is out there.
Coming back to your age defence then, and the things that we know are starting to work and do well, like the DNA repair. Once you've got that in the product, how does it actually work? How does it then identify what it needs to fix?
You know, I said that the interest in DNA repair enzymes has been around for a long time, but actually getting them into these little packages, so liposomes are like that, they're kind of like little packages, like your exosomes, but the research in developing DNA repair enzymes in such a way that they could transverse the stratum corneum and get into the cells where they needed was the hard work, basically. And that was the work that Daniel Roche and his team were doing for years, trying to figure out how could they get these enzymes into the skin, because they knew that they could work in the skin, but how could they get them in there? And the way that they figured out how to do it was to package them in these little liposomes. So they are encapsulated in a way that they can transverse the stratum corneum and get into the cells where they're needed. And the two that we use, there's one called photolyase, which is the one that's activated by light, and then another one called endonuclease. So the photolyase actually can repair the damaged DNA. The endonuclease basically snips out the damage and then lets your body's own repair processes come along and repair the DNA. So, it's all really fascinating stuff. I think one of the reasons why DNA repair enzymes kind of haven't taken off in the same way as antioxidants have is that from our perspective as dermatologists, it really, really makes sense because of the way we see photoaging and photo damage and how abnormal our DNA damage has a major impact on that. But when you are talking about cosmetic products, you're thinking about things like collagen and wrinkles. And so actually explaining to people that DNA repair makes sense for your skin in the long run is quite a difficult message to get across. And I think that's probably why, I mean, even the antioxidants were quite a difficult message, but I think people really understand antioxidants now. But I think that with the DNA repair, we have a bit more work to do in explaining why they are beneficial in your skincare and why you really should be using them.
To me, it just sounds like a no-brainer. I mean, you gave me a little bit to try before, so can I expect to wake up tomorrow morning with baby hands, or does this take quite some time?
I mean, so basically it is repairing immediate DNA damage. It's not repairing damage that was done or mutations that were done years ago. However, in repairing the immediate damage, you are reducing inflammation, you are creating a better environment for all of the other processes that we talked about in your skin, because your body isn't in this kind of yin-yang fight-flight mode trying to repair all of the new damage. So everything that is reducing all of that cumulative photo damage as the years go by is really, really beneficial for your skin in the long run, basically.
Are there any risks associated with them?
Not that I'm aware
No,
of.
I mean, they are very, very direct to just acting where they are told. They will only act on the damaged UV-induced DNA breaks in your skin and nowhere else. And when they get into your skin, I mean, a lot of the information I know about, I've read obviously a lot of research papers on DNA repair enzymes, but really Daniel Roche is kind of still considered the world expert because he spent his life studying them. And anything that I know from him and my communications with him. So Dan said that basically once they get into your skin, they're probably there for about 24 hours and then they've done their job.
So this is something that you really do need in a daily skincare routine.
But interestingly, the little enzymes, so say take the photolyase, it sits on the little area of DNA damage. Photon of light comes along, it repairs it, then it goes off and finds another area of DNA damage. It doesn't just lose its ability to continue to repair with that one repair.
Does that mean that you have to be lying out in the sun 24/7 for it to do this?
Oh, no, please don't do that. It's just,
I'm going to.
no.
It's photo reactivation, isn't it? Not UV.
Yeah, so it's photo reactivation, so it needs visible light.
Speaking of visible light and those sorts of things, the other thing, apart from just UV, a lot of people now want to make sure that their products have, the blue light from computers and screens and things like that, and other wavelengths of light. Are they damaging us, or is this a marketing thing?
The sun is a source of electromagnetic radiation, and the shorter wavelengths are your UV wavelengths, and then visible light is the next sort of zone in the electromagnetic spectrum. So blue light is the closest, or the violet blue kind of colour is very, very close to UVA, and obviously it's a spectrum. So because it's so close to UVA, it has some UVA-like effects. So the things that UVA does, which is penetrate deep into skin and cause oxidative stress, the blue light or kind of high energy visible light, it can do similar things essentially, because electromagnetic spectrum is a spectrum. That's essentially why it does that.
So does your traditional sunscreen then protect against those, or do you have to make sure it's got another filter in it then?
If you think about the physics of light, things that block light are things that have colour in it. So in order to block blue light, you need something with colour in it. So a tinted sunscreen or foundation or anything which has a tint, because if you cannot see someone's skin, that blue light is also not getting into the skin. So you can just have that as a rule of thumb in terms of blocking blue light. The other thing to say is the vast majority of blue light exposure that we're getting is from the sun.
Yes,
so the sun
not from your screens.
The sun is a massive source of electromagnetic radiation. Obviously, we have lots of benefits from being out in the sun. I cannot emphasize enough. There's a lot of epidemiological data showing that being outdoors has health benefits, but it comes at a cost to your skin.
Yeah.
And it's about balancing those two things and the risks and benefits. And we can attenuate those risks by taking some simple protective measures. That includes modifying our behaviours, as well as using some protective clothing, as well as using some protective products. So going back to the blue light thing, there are sunscreen filters which can extend into the blue light spectrum, actually. So
so usually up to like 400 nanometers, and then you're getting into that high energy visible light. Unlike the SPF value and the PA+++ value, or the little circle around the PA, that's all regulated. With blue light and infrared and things like that, that's a different category. So a lot of actually what companies use are antioxidants, because blue light and infrared and all of those other areas of electromagnetic radiation cause reactive oxygen species and free radicals, and they are basically dealt with through the antioxidants.
You mentioned other things about the sun that are good, vitamin D. So the other thing that people worry about then is that, well, if I'm covered from head to toe in sunscreen, am I going to be getting enough vitamin D?
So vitamin D is a hormone that is very important for our bodily functions, and the main sources of vitamin D are through cutaneous synthesis from UV exposure and also through our diet. And vitamin D biology is really, really complicated, and I think it's often oversimplified and misunderstood. So people think that if they just lay out in the sun, they will get more and more and more and more vitamin D, and then they will be supercharged with vitamin D and immune to disease. That seems to be a concept that is out there in social media.
So
would you agree?
Do it, Fiona. Yes.
But that is not true. That isn't what happens. So first of all, your vitamin D synthesis in your skin, it does reach a plateau. So after a certain point, you will not be making more vitamin D. And obviously that is influenced by things such as your skin tone. So if you have more deeply pigmented skin, you are less efficient at making vitamin D in low UV environments, and sunscreen can reduce it. But we know that in real world application, actually the amount of UV exposure required to generate vitamin D is actually not that much. And people's use of sunscreen doesn't seem to affect how much vitamin D they are making. And the other thing to take into account is that once you've made that vitamin D and also consume some vitamin D through your diet, your body is then processing the vitamin D through very complicated mechanisms that involve your liver, your kidneys, etc., and the bioavailability of your vitamin D is also something that's influenced by a lot of other factors. So it's not as straightforward as just sitting out in the sun and then you will boost your vitamin D and you are immune to all diseases. It's a lot more complicated than that, and I think this is a really ongoing active area of research. If you go back 70 years, doctors were actually recommending that people purchase sun lamps to make vitamin D at home on their skin and recommending tanning. And the thought process is really reversed because it wasn't understood then the negative impacts of having that much UV exposure. And really I think that the benefits of sort of overdosing and using sun exposure to generate vitamin D is, I think that's really an overstated aspect of vitamin D biology, and I think it's really not very well understood. So what your level of vitamin D is in your body is not just influenced by how much sun you're getting, it's influenced by lots of other factors in your body and your health status. So if you look at the sort of vitamin D body of research, if you look at epidemiological studies, you'll come across dozens of studies saying low vitamin D is associated with every single bad thing out there. Heart disease, dementia, cancer, worse cancer prognosis, all of those kinds of things. But giving people vitamin D never seems to actually reverse those things. So there is a
study about, there was a very big study, I can't remember what it was called, that was published, that it was like an all-cause mortality study where people took half the cohort took vitamin D supplementation and then half didn't, and they've realised that actually there wasn't a reduction in all-cause mortality or cardiovascular risk, which was one of the major things. So we still have a lot to learn about vitamin D. I think if you're worried about your vitamin D levels, you can get them checked. It's very easy to get vitamin D supplementation. It's an easy little spray. One of the things that's interesting now that seems to be coming up in skincare are vitamin D receptors that, again, myself and Beibei were just talking about a study. I must show you that one because I have it now. A study on vitamin D receptors in the skin. So that's another fascinating area for skincare science.
In what way?
So it looks like UV overexposure can cause harm to your vitamin D receptors, in that oxidization basically can cause harm to lots of things. It can also cause harm to your vitamin D receptors. And if you reduce oxidization in your skin through things like antioxidants, then your vitamin D receptors will function better.
Oh, okay. And if they're damaged,
This-
is there a point where your vitamin D receptor gets so damaged that it just doesn't work anymore, or do we just not know enough
we
about
don't
it?
know.
So there are studies where they've looked at how much vitamin D is generated by someone exposed to a dose of UV at different ages, so older people versus younger people, and older people do not make as much vitamin D with the same dose of UV exposure. So there is an aging effect in terms of vitamin D synthesis in the skin. But like I said, this is a really complicated issue, and I think it's really oversimplified. People love the idea that the sun is just giving us health. But I think that is only partially true and only to a limited extent when it comes to the vitamin D story. And then you have to think about your skin longevity and the impact of that UV on your skin longevity. So you don't need to get burnt, get deeply tanned to gain adequate vitamin D levels. The recommendation is that if you have a bit of sun just on your forearms for 10 to 15 minutes, that is adequate. If you have a lighter skin tone, you might need a slightly more if you have a darker skin tone, but that is the general recommendation. So I think it's just something to have a balanced view on, rather than being sort of too polarized in one direction.
Yeah, it's an interesting one, isn't it, too, especially with supplementation, because people, there's often that sort of thought, well, if a little bit is good, then more must be better. And I've come across some people who are saying they would take, who'd not ever had a diagnosis of a deficiency, saying they were taking 20,000 international units a day, and you're thinking, this is a fat soluble vitamin. You just what are you doing? Because there are risks with too much of anything as well.
Yeah, absolutely. And people can get toxicity from overdosing vitamins. And another one that people can develop toxicity from is vitamin A. So vitamin A toxicity is something that has come up quite a lot recently as well, because you can get some interesting skin manifestations from vitamin A toxicity. Your skin can actually turn slightly orange. And you can even get it from eating too much vitamin A rich foods. So you can definitely have too much of a good thing. And it's just, I think, definitely following very basic nutrition guidelines is actually not a bad place to start.
Yeah.
Absolutely. And basic skincare guidelines as well.
And basic skincare,
Absolutely. Ladies, thank you so much for that. That was riveting.
Thanks, Fiona.
Thank you.